So today, we're going to tackle something really cool, demystifying how you actually build a high-quality genome from the ground up. We're talking about going from a physical, biological sample all the way to a complete digital blueprint of an organism's life. It's honestly wild to think how much has changed. I mean, just 20 years ago, sequencing even one complex genome was this massive international project. It literally took a global effort, a kind of scientific moonshot, for every single species. Well, that world is gone. Thanks to some incredible revolutions in sequencing tech and bioinformatics, this kind of power is now in the hands of individual labs, even students. Suddenly, any organism can become a model organism. We can unlock the biological secrets of, well, pretty much anything. Okay, so that's the big question, right? How do you actually do it? What does it really take to go from, say, a leaf clipping or a tissue sample to a fully annotated chromosome-level genome assembly? Let's walk through the whole process step by step. And it's so important to get why this matters. A reference genome isn't just a long string of A's, T's, C's, and G's. It's the foundation. It's the cornerstone for everything that comes next. Powerful studies looking at disease, evolution, and the very architecture of life itself. All right, first up, phase one. And this is all about the blueprint. Before you even think about sequencing a single molecule of DNA, you have to have a rock-solid plan. This is where you make the big, project-defining decisions. And the first huge choice is your sequencing technology. It really boils down to a classic trade-off. You've got short-read tech on one side, it's super accurate, it's cheap, but it chops the genome into millions of tiny little pieces. Trying to assemble a genome with just short reads is like piecing together a novel that's been put through a paper shredder. It's almost impossible in the tricky, repetitive parts. Then you have long-read technologies. These are like recovering entire paragraphs or even pages at a time. They just sail right over those complicated regions and solve the puzzle. It's how we finally mapped human centromeres, unlocking parts of our DNA that were basically dark matter for decades. Now, the trade-off used to be cost and error rates, but that gap is closing fast. You know, what's funny is that you can have the fanciest machines and the cleverest software in the world, but none of it can save a bad sample. The success of the entire project, and I mean all of it, hinges on getting really clean, high molecular weight DNA. That means no contaminants and, crucially, making sure the DNA strands are long and not broken into tiny bits. It's the absolute golden rule of genomics. Garbage in, garbage out. And of course, there's more prep work. You've got to do your homework on the organism. How big is the genome? Is it diploid, with 2 copies of each chromosome, or something way more complex? How much repetitive junk DNA are you going to have to wade through? To make your life way, way easier, you should try to use a single inbred individual. It just cuts down on the genetic complexity you have to sort out later. And finally, don't try to go it alone. Building a team or a consortium helps pool money, knowledge, and resources. It turns a huge task into a collaborative effort. So, with our blueprint drafted and our precious data in hand, we move into phase 2, the digital build. This is where the real magic happens. It's where we take all that raw sequencing data and start turning it into a coherent biological story. Okay, I know what you're thinking. This flowchart looks intense. And yeah, it is. It's our map for the whole digital process, showing all the different paths you can take. But don't worry, we can actually boil this whole complicated workflow down into 4 essential core steps. At its heart, the process is this. First, you assemble. You're digitally stitching together millions of reads into longer pieces we call contigs. Then you polish. You use the raw data to go back and fix any mistakes in that first draft. Next, you assess the quality to see how good of a job you did. And finally, the really fascinating part, you annotate. That's where you figure out where the genes are and what they actually do. So how do we measure that quality? Well, one of the key stats is something called N50. Think of it like this. Would you rather have a book broken into 10 long, complete chapters or 10,000 tiny scraps of paper? A high N50 value means your genome is assembled into big, useful chunks, more like chapters and less like confetti. It tells you your assembly isn't hopelessly fragmented. Any annotation phase? It's got its own amazing digital toolkit. You'll use programs like Maker or Breaker to predict where the genes are. You'll use Repeat Masker to find all that repetitive DNA. Tools like Blast2Go help you figure out what those predicted genes might actually do. And then you get to visualize the whole shebang in a genome browser, which lets you literally fly through the chromosomes and explore the data. It's incredible. And that brings us to the final and, maybe the most important phase of all, The goal here isn't to create some static file that just sits on a server. It's to create a living, breathing scientific resource that can serve an entire community of researchers.
Yeah, this is the crucial takeaway. A genome assembly is never truly finished. It's really just a hypothesis of that organism's genetic code. And as we get new data, better software, and a deeper understanding, that assembly and its annotation have to be updated and refined. It's a data set that evolves right along with our own knowledge.
So, how do we make sure all this hard work pays off for years to come? That's where the FAIR principles come in. The data has to be findable, accessible, interoperable, and reusable. This isn't just about being tidy, it's a whole philosophy that ensures the massive effort of sequencing a genome benefits scientists all over the world, not just the original team. And this is where it all comes together so brilliantly. Web portals allow a whole community of researchers, each with their own specialized knowledge, to log in and work together to improve the annotation. An expert on one gene family can fix its structure. Someone else can add functional data. And together, they build a far more accurate and valuable resource than any single lab ever could on its own. Okay, we've covered a lot of ground. So let's pull all this theory together and build a concrete step-by-step pipeline. A recipe, really, for anyone looking to start their first genome project right now. Today, a really powerful, proven workflow looks something like this. You start with pure long reads. Then you assemble them into contigs with a tool like Fly. You check your quality and completeness with Busco, which makes sure all the essential genes are there. Then comes the real game changer. You use high-C data to scaffold. This tech actually tells you which bits of the genome are physically close to each other inside the nucleus, letting you stitch your contigs into full chromosomes. After polishing out any last errors, you can kick off the annotation. It's a modern, powerful, and proven approach. So, that's the roadmap. The tools, the techniques. They're more accessible than they have ever been, turning almost any organism on Earth into an open book just waiting to be read. Which brings me to the final question for you. If you could pick any species, what would you sequence, and what secrets do you think its genome is just waiting to unlock?